Hello, dear students. We'll start today with the group of diverse microorganisms belongs to the category of gram nectar rods. And as we all know that they're called rods because they are long in shape. They are stained gram negative when they are put under the gram stains procedures. Now, this large group of microorganisms, it can be, categorized into three main groups based on to their location in the human bodies. Some of the members of this gram-negative organism group, they affect mostly the enteric tract, and some of them they have effect on the respiratory tract and other are zoonotic in nature. Now, within the enteric tract, there are some genera whose members primarily affect both within and outside the enteric tract, and this includes the members of the genus Escherichia and Salmonella. Some organisms belong to the genus Shigella, Vibrio, Campylobacter, and Helicobacter. They affect both primarily within the enteric tract, and some outside the enteric tract. That includes the Klebsiella Enterobacter ratio group. Then we have the Proteus, Providentia, Morganella group, the Pseudomonas, and Bacteroids. And we have already done through the respiratory tract organisms like Haemophilus, influenza, Legionella pneumophila, Bacteria tucis, and of course the zoonotic, which are transmissible from animals to humans, including the genera Brucella, Francisella, Pasteur, and Yersinia. We will talk about the diseases which are caused by the gram-negative microorganism, and these diseases, they are related to the enteric tract. And if you look at that, we have three important genera that are frequently pathogens in the enteric tract of the human and animals: the Salmonella, the Shigella, and the Campylobacter. And the less frequent pathogens in the enteric tract are the Escherichia, Vibrio, and Yersinia. The Salmonella, Shigella, and Campylobacter and Rickettsia, they are also associated with high incidence of autoimmune diseases like the reactive arthritis and Ritter syndromes. Now, the reactive arthritis is actually, you see the pain in the joints and swelling in the joints, particularly of knee and ankle. And they're triggered by the infections in the other part of the body, mostly the intestinal tract, the genital tract, the urinary tract infections. The Ritter syndrome is, you see, it is the formally referred to be as what we call as the Ritter syndrome. The reactive arthritis is technically or formally it is referred to be as the Ritter syndrome, and it is more, as you see, it can involve the eyes also, it can involve the ears also, and all that. The UTI infections are the organism of the gram-negative rods that affect the urinary tract. They include the Escherichia, and of course the less frequent microorganisms are the Enterobacter, Klebsiella, Proteus, and Pseudomonas. So the gram-negative rods, the major group of these organisms, they affect the enteric tract, and we have three important genera: the Campylobacter, the Shigella, and of course the Salmonella. You see, there is the EnteroBactriaceae, as we all know, is a large family of the gram-negative rods, which is primarily present in the colon of both animals and humans. They occur as a normal flora of the human body. These organisms, they are major facultative anaerobes in the large intestine. Means that these organisms have the potential to grow both in the what you call as they can synthesize their ATPs or energy both through the fermentation and oxidation process. And these organisms, they're relatively small in numbers in the colon compared to that of the strict anaerobes, which are the bacteroids. Bacteroids predominant in the colon compared to that of the members of the gram-negative rods or Enterobactriaceae family. The features which are common to all members include the anatomical location and the other four metabolic processes which are very important for the identification or for the categorization of these microorganisms. There are four metabolic reactions that are very, you see, peculiar or important with these gram-negative rods. All gram-negative rods, they are facultative anaerobes. They are facultative anaerobes. All gram-negative rods, they ferment glucose, particularly the members of the family Enterobactriaceae. We can strictly confine our discussion on to the Enterobactriaceae family. This is characteristic. All members of the family Enterobactriaceae, they are facultative anaerobes. They ferment glucose, all of them. But the fermentation of the other sugars like lactose, like sucrose, that varies. It varies with reference to different organisms. But all of them, they ferment glucose. None of them has, you see, the cytochrome oxidase enzyme, but that's why we call it oxidase negative. Majority of them, they are oxidase negative. And they all have the ability to reduce nitrate into nitrites and as a part of the energy generation processes. So we are mainly in the Enterobactriaceae. These members of these groups, this family, they are facultative anaerobic rods that ferment glucose, they are oxidase negative, and they reduces nitrates to nitrite. Now, in this table, we will comprehend the diseases caused by the member of the Enterobactriaceae. And if you look that we have almost 1, 2, 3, 4, 5, 6, 7, 8 important genera. The major pathogens of the family of the Enterobactriaceae that belongs to these major genera are the genera of Escherichia, the Salmonella, Shigella, Klebsiella, Enterobacter, Serratia, Proteus, and Yersinia. Now, if you look at the Escherichia, the representative diseases, the UTI infection is very common. Very, very common with the UTI, with the Escherichia, particularly the Escherichia coli. It also causes traveler's diarrhea, and it is one of the most two important organisms that causes neonatal meningitis. So the Escherichia, it causes neonatal meningitis, it causes traveler's diarrhea, and it causes UTI infection. Now, the foremost common cause of the neonatal meningitis is Neisseria meningitidis, and this is the second most important organism. Shigella is responsible for the dysentery, which we all know it's a bloody diarrhea. The Salmonella, it causes, it has many organisms. It has more than what you call as 2,200 serovars or serotypes. And we have the species like Salmonella typhi, Salmonella paratyphi, causing, you see, the typhoid fever or enteric fever respectively. And we have non-motile salmonella, what we call as the non-typhoidal salmonella. They cause the entitis, what we call as the enterocolitis. The less important genera which are related with these, they are the Arizona, the Cetobacter, and Adventitia. Now we have another important group of microorganisms belong to the genus Klebsiella, and the important species, the Klebsiella pneumoniae, it causes pneumonia and again the UTI infections. Enterobacter, we have the Enterobacter colici that causes pneumonia, UTI infections, Serratia, and we have the Proteus, and we have the Yersinia. We have covered all these. So these are the 7 major genera of the family Enterobactriaceae. Now, the pathogenesis of the members of this family Enterobactriaceae. Now, as we know that these are gram-negative rods, so if you recall your memory regarding the anatomical structure of the gram-negative rod bacteria, they have the flagella. They have a long flagella, they have the pili, and some of them they have the capsules, they have the cell wall and the cell membranes, and of course these are the very standard. So all members, because they're gram-negative rods, they have what we call as the lipopolysaccharide layer, and this lipopolysaccharide is of course the endotoxins. All members of the family Enterobactriaceae, they contain endotoxins. Now, apart from the endotoxins, they produce several exotoxins. And the exotoxins produced by the members of the family Enterobactriaceae, they are referred to be as enterotoxin. The Escherichia coli toxin, which act like a Shigella toxin, the Shigella toxin. The Vibrio cholera toxins, which we call as the choleragen, they are the exotoxins. So members, they produce exotoxins as well as they produce the endotoxins. And now these two toxins, the enterotoxins or the exotoxins of members of the family Enterobacteriaceae, they act by activating the adenylyl cyclase within the cells of the small intestine, leading to the diarrhea. Now we have a specific stain of the Escherichia coli, what we call as the O157. This is a stain which belongs to the somatic antigen. We will discuss it right now. And they produce Sajid-like toxin, and it causes hemolytic uremic syndrome. Hemolytic uremic syndrome caused by E. coli O157. We will discuss this organism in detail. So the pathogenesis of the members of the family Enterobacteriaceae is basically because of the production of endotoxins. They do produce exotoxins, and they causes diarrhea, and they have some of them they have the capsules, and they have the villi that help them to adhere to the intestinal mucosa, and later on penetration and release of toxins and the production of enterocolitic signs and symptoms. Now, the antigens of this group of microorganisms, they're very important. Particularly, they're very important with respect to the family of Salmonella and Shigella. We have several members of the family, as we have seen, but the antigens belong to the genus Salmonella and Shigella. They are very, very important. These antigens of Salmonella and Shigella, they are used for the identification of both the clinical laboratory and in the epidemiological investigations. So in order to find identification of the microorganism belonging to the genus Salmonella or the genus Shigella, we have to go for the identification with the help of the different antigens. Now, there are three important surface antigens. Now, because the organism, they have the cell wall, so we have the cell wall antigen, what we call as the somatic antigen. It is referred to be as O antigen. Now, we have these organisms, they have the flagella, so we have a flagellar antigen, which is referred to be as H antigen. And you see, not all the species, they have the flagellar antigens. Only those species which are motile with the help of flagella, and they do have the flagella, they have the flagellar antigens. We have the capsular, which is the polysaccharide antigen, and not all species of the families of Enterobacteriaceae genera, they have the capsular antigen. Only those species like Klebsiella pneumoniae. It has a very, very, you see the excellent capsule. So the members of the family which have the capsules, they have the capsular antigen. Those which have the flagella, they have the flagellar antigen, and all of them they have the cell wall, so they have the cell wall antigen, what we call as the somatic antigen or O antigen. All right. Now, how do we generally diagnose the infections with the Enterobacteriaceae family or the gram-negative family, right? So this is generalized lab diagnosis regardless of the genera of the species. The protocol that we can adopt or follow to isolate and identify the gram-negative rods, including the family Enterobacteriaceae genera. Now, normally we in the laboratory, in the diagnostic microbiology laboratory, we use two media. One, as the standard, is the blood agar. And other one, it has to be a selective differential medium. And we all know that we have very important medium, which is selective as well as differential, particularly for the family of Enterobacteriaceae and gram-negative rods, and we call that McConkey agar. And we have another one which is known as the EMB agar, what we call as the eosine methylene blue agar. We have the dye incorporated in this medium as an inhibitor for the organism, for the gram-positive rods. So always you see it is a standard procedure to culture this specimen onto two medium, at least on two medium. One is the blood agar, and the other one may be a differential selective medium. Maybe it is a McConkey agar, and maybe you can use the eosine methylene blue agar. So keep in mind, first the isolation of the organism by sticking the sample onto the blood agar along with that of the one of the differential selective medium, either McConkey agar or eosine methylene agar. Now, the differential ability is based onto the lactose fermentation. Now, which is the specific microorganism? So, after culturing the microorganism onto one selective medium along with that of the one enriched medium, we will see the lactose fermentation test. And this is the most important metabolic criterion for the differentiation of the species of the family Enterobacteriaceae. growth of the microorganism, then lactose fermentation test. And based on to the fermentation or non-fermentation of the lactose, we have two main groups. We have the non-lactose fermenters and we have the lactose fermenters. The non-lactose fermenters include the members of the family Salmonella and Shigella, whereas the lactose fermenters are the Escherichia coli. So, broadly speaking, the gram-negative rods are the Enterobactriaceae family general, maybe lactose fermenters, non-lactose fermenters. Lactose fermenters are the Escherichia, like the Escherichia coli. Non-lactose fermenters are the Salmonella enteritidis, Shigella dysentery. Now, the selective action effect is supporting unwanted gram-negative dyes, the dyes that has been incorporated in the McConkie agar or in the EMB agar, they are the bacteriostatic. It is incorporated with the bile salt and dyes, which are the bacteriostatic. So always remember to culture the microorganism or the sample onto 2 medium and then go for the biochemical fermentation test. And based on to the lactose fermentation, we have lactose fermenters and lactose non-fermenters. Now here this how the growth of different genera of the family Enterobactriaceae or gram-negative rods will look like onto the McConkie agar. Now if you look at this, this is the lactose fermenters colonies. The colony, the organism has fermented lactose, and this lactose fermentation has resulted into the acid production, and this acid production have, you see, has effect on the dye in that, and give it a pink color. And the non-lactose fermenters colonies, they are the colorless. So we know that the lactose fermenters are the Escherichia colony, whereas the non-lactose fermenters are the colony which may be of Salmonella or maybe of Shigella. Now, here you can also see they are various organisms, but keep your concentration on this slide. If you grow the microorganism or a sample onto a McConkie agar and you want to distinguish the colonies of Escherichia coli with that of the Salmonella, I mean the lactose fermenters colonies of Escherichia coli from non-lactose fermenter colonies of the Salmonella or Shigella, this is how they will look like. And we will go into the mechanics of this colony characteristics, that how they appears. Now, similarly, you see if you have a sample that has been staked onto the eosinomethylene BU agar, the Escherichia coli colonies will provide green discoloration. The green sheen is produced on the EMB agar. The E. coli colonies would appear like green sheen behind that, whereas the rest of the colonies, they are different in color. So they are the lactose fermenters, they are the non-lactose fermenters, right? So this is one criteria. Now the other one is that, now we see that, what are the lactose fermenters and non-fermenters? The lactose fermentation occurs in Escherichia, in Klebsiella, in Enterobacter. So members of the genus Escherichia, the Escherichia, Klebsiella, and Enterobacter, they are the lactose fermenters. They will give you like these colors of the colonies. Now those which does not cause lactose fermentation, the Salmonella, the Shigella, the Proteus, and Pseudomonas, they will look like this. But some of the colonies of the genus Serratia and Vibrio, they are late fermenters, and this very slowly cause fermentation, maybe at later stage, after 1 week's time, 10 days time, 12 days time, you may have some of the colonies which are of this color, is what we call it, this Serratia. So based on the fermentation, we can really distinguish the group of the microorganism on the McConkie agar and onto the eosinomethylene agar. Now, in addition to the eosinomethylene agar or McConkie agar, we have to use another medium, what we call as the TSI agar, the triple sugar iron agars. And as we know that we make a slant in this case, that instead of putting the medium on the battery plate, we put them in the test tube and then we tilt that test tube at an angle of 44 degrees centigrade and we make a slant. We made a slant. These are the TSI slants. So it has two components. This is up till here, from here till here. This is what we call as the butt of the medium. And from here to here, it is the slant of the medium. Now this area is poorly oxygenated up till here. This area is highly oxygenated and it has the great you see significance. Just keep in mind. Now, the triple sugar iron, it contains three sugars: the glucose, the lactose, and sucrose. It also has a phenyl red as an indicator, and it has a ferrous sulfate, the ferrous salt, which are used for detection of the ferrous sulfide. It gives you the black color of the organism. Sorry for that. Now, if you look at the composition, the glucose is just 0.1%, whereas the lactose and the sucrose are about 1%. This means that glucose is 10 times less than that of the concentration of lactose and sucrose. Or in other words, we can say that the lactose and sucrose, they are 10 times more in concentration than that of the glucose. So we have three sugars: glucose, lactose, sucrose, a phenylalanine indicator, and a ferrous sulfate that is used to see the conversion of the ferrous sulfate into ferrous sulfide. Now, the interpretation of this test, we can make that if the lactose or sucrose is fermented, I mean the high concentration sugars with a 1% of the medium, a large amount of acid is produced. And this turns the phenol red indicator yellow. A large amount is produced, and the phenol red indicator, it is turned into yellow. Phenol red indicator is turned into yellow, both in the slant and in the bud. You see, like this, entire area. This means that both lactose and sucrose has been fermented, and the entire bud and slant, they are yellow color because of the fermentation of this. Now, if you look at this, this is because of the fermentation of gas. The gas bubble has pressurized the bud, and the bud has raised from the base. So this is you see, this is fermentation of both lactose and sucrose with the production of gas. This is with the production of gas. We can see over here. Now, if lactose is not fermented, but the small amount of glucose, which is just 0.1%, it is fermented, the oxygen deficient bud, you see, there's no oxygen, less oxygen, and there's more oxygen. The oxygen deficient bud will be yellow, but the slant will be acidic. This will be yellow, this will be acidic in some area. And there will be production of the conversion of the ferrous sulfide straight into sulfide. You will have the black color of the organism. So this is how to just make you understand that what is the composition of the TSI. This is an integral component of the diagnostic procedure that we adopt for the identification of the members of the family Enterobactriaceae. Grow them on one or two of the differential medium, and after that, when you have the colonies, you make a stab onto the base of the medium, what you call as the bud, and then here, and then you stick onto the surface, and you can have these kind of reactions. Culture the organism on the differential medium, and then take the colony, put it onto the TSI agar slant, and see the behavior of the colonies. And then you go for the biochemical fermentation test, like lactose fermentation, as you can see over. This is a biochemical fermentation test, actually. The TSI agar slant is actually a biochemical fermentation test. And then we identify the microorganism based on to the antigens, the somatic antigen, the flagellar antigen, the capsular antigen, or in case of Salmonella typhi, we go for the virulence antigen, what we call it the VI antigen. So different organisms, they have different behavior. This is how the different organisms, they can have the, here you see, we can have all sugars fermented. This is, you see the slant is fermented with the production of, you see the blackening of the colony, H stress is produced. You see, none of the sugar has been fermented. Gas produced has been there. So various organisms, they produce various discoloration of the slant medium. This is how the TSI agar is used. Now, broadly speaking, that what is the reaction of the different organism onto the TSI agar? Now, as we know that we have a slant in the TSI agar, we have a bud, and maybe there is a production of gas, or may not be a production of gas. There is a production of estrus, or maybe not be a production of estrus. And what are the representative genera? The Ascracia Enterobacter, and Klebsiella. They make acidic slant and acidic butt. Means that entire slanted butt are yellow in color. They may produce gas or they will not produce the H2S. There will be no blackening of the medium. Entire butt and slant would be, you see, the yellow in color, and there will be a production of gas. Now, in case of Shigella and Serratia, no gas production, no H2S production, alkaline slant and acidic butt. Alkaline slant means that slant would be of pink in color and the butt would be of yellow in color. There will be no gas, there will be no H2S, so we can suspect as Shigella. Now, in Salmonella and Proteus, there will be a blackening of this, the butt or maybe of the slant. There will be a gas production. Now, the butt would be, you see, the acidic in color. The slant would be alkaline, so similar to that of the Shigella, but in both cases, the gas and H2S is there. The Pseudomonas, negative, entire alkaline. So if you see the entire, there is no gas production, there is no blackening of the butt or slant, and there is the both alkaline and slant are alkaline, so we can go for the Pseudomonas. So keeping in view these characteristics, we can really identify the genera of the microorganism or the triple sugar iron, which is a biochemical test reactions. Now, coliforms do have their role to play in the public health also. What is the relationship between the coliforms and the public health? They contaminate the public water supplies. The coliforms, they contaminate the public water supplies by the sewerages. The sewerage, they are full of coliforms, and when this sewage supply, they contaminate the water supplies, then the disease problem occur, and it create a lot of public health issues. The presence of the coliform, it is detected. I mean, we can have, you see the Enterobacter, Klebsiella also, but if there is only the presence of coliform, like the Escherichia coli, this is an indicator that it is a fecal contamination. So presence of Escherichia coli in drinking water is a clear indication that the drinking water has been contaminated by the fecal material or sewage, right? But if there is Enterobacter or Klebsiella, it may have been some other source. But if there is presence of the Escherichia coli, because it is exclusively found in the sewage, that is clear indication that the water supply has been contaminated with the sewage water. So the water quality testing, we always go for the determination of the Escherichia coli. If we have been able to isolate and identify a specific number of Escherichia coli cells in the water, this means that the water quality is good or poor. The water quality is based on to the numerical value of the Escherichia coli. It's good or bad. Now, these organisms, they ferment lactose with the production of a certain gas. They can grow at 44.5 degrees centigrade, and they can produce characteristic colony onto the MB agar, like the green discoloration as I told you. The clonicore count is about 4 per deciliter in municipal drinking water, indicative of unacceptable fecal contamination. If you have less than 4, 1, 2, the water is acceptable for drinking. But if you have more than that, the water is not acceptable for drinking purpose. These are the enteropathogens that are killed by the chlorination of the drinking water, but sometimes they do resist. So the Escherichia coli, it does have what you call as the public health signification. It is significant because of public health issue. If contaminated with a large number of Escherichia coli, it is an indication that it has been adulterated with the sewage water. So we have to be very, very careful. Now that was a generalized concept that we have to deal with in the gram-negative rods or in the ENTEROBACTRIACEAE. Now we deal with this specific microorganism. But before we do that, let's have a generalized idea that how we can treat these infections of ENTEROBACTRIACEAE genera, or the members of genera of the ENTEROBACTRIACEAE, like Salmonella, Shigella, Escherichia, Proteus, Klebsiella. The treatment is, you see, it is tailored individually to antibiotic sensitivity of the organism. Especially the UTI infection, it is very, very these organisms are very, very resistant. So we will have to adopt our strategies. I will have to tailor our strategies, our procedures to the antimicrobial sensitivity testing. A wide range of antimicrobial drugs are effective, and they're really effective, starting from penicillin, cephalosporins, aminoglycosides, chloramphenicol, tetracyclines, quinolones, sulfonamides. So a huge group of microorganisms and a huge variety of drugs that are available to counter with these microorganisms. The enterograminator they are highly drug resistant because due to the production of beta-lactamases, and they have the drug modifying enzymes also. So because of the production of high amount of beta-lactamases, and because of the modification of the drug receptor sites by the enzymes that changes these receptors, these organisms, they are highly resistant, particularly to the enteric rot. These organisms, they acquire drug resistance mainly because of the conjugation. They acquired the resistant plasmid that we have seen in the previous lectures when we were studying the intermicrobial genetic recombination, and we went for the conjugation, transformation, transduction, and in this way they have the multiple drug resistance. So plasmid-mediated drug resistance is very common in the genus of the family ENTEROBACTRIACEAE. Now, that was the generalized idea that what are the graminator rods and what are the important genus of the family ENTEROBACTRIACEAE, where do they act, how do they act, what kind of disease they produce, what are their antigens, what antimicrobial chemotherapy we can use to kill these microorganisms or to get rid of microorganisms. Now we'll move on to the specific microorganism, and we will discuss the genus whose organism or the members of whose they really cause what you call as diarrhea. And these organisms, they have flagella, they are elongated in shape, and you understand they cause one of the very important disease what we called as the diarrhea. And specifically speaking, the group of this microorganism is related with the traveler's diarrhea. It does cause UTI infection. The members of this genus does cause UTI infection, does cause sepsis, but they are also responsible for the traveler's diarrhea. And we are going to discuss the group of microorganisms belong to the genus Escherichia. Very common name, very common species. Once you may have been in the 10th grade class or in the first or second grade class, you must have studied or you must have at least aware with the name of this microorganism. Escherichia is the name of the genus, and we have many species. Now, Escherichia are the most common cause of UTI and gram-negative rod sepsis. We have many organisms that cause sepsis. Some of them are gram-positive. But from within the gram-negative, Escherichia are responsible for the sepsis. They are responsible for the urinary tract infections. And one of the two important causes that cause a neonatal meningitis is the Escherichia. So look at this. This organism does not only frequently associated with the traveler's diarrhea, of course. The traveler's diarrhea, the individuals who travel in the ships, cruise, and they get this infection. And this diarrhea, the traveler's diarrhea, which is the blood diarrhea, it is an enterohemorrhagic diarrhea. It causes, you see, the hemorrhages and all that. And its specific lysogenized stain is responsible for the production of enterohemorrhagic stain, which is responsible for the bloody diarrhea. So organism causes, you see, UTI infections. It causes sepsis. One of the second most important cause of the neonatal meningitis. Traveler diarrhea is very common, and not all strains of Escherichia they cause traveler diarrhea. Only the lysogenized stains that has been infected with the phages and transferred the genes to this mechanism, they causes the bloody diarrhea. And the stains of Escherichia coli that causes bloody diarrhea, traveler's diarrhea, we call them the enterohemorrhagic stains. Enterohemorrhagic stains of Escherichia coli are responsible for the bloody diarrhea. Now, important organism, these properties of the gram-negative Escherichia coli, This is the most abundant facultative or anaerobic in the chlone. You see, the feces is the feces is greatly outnumbered by the bacteria. In 1 gram of fecal material, we have billions of cells of the bacteria. But after bacteria, which are the strict anaerobes, the facultative anaerobes encompasses in the colon and the feces by the Escherichia coli. Now, these organisms, they ferment lactose. So as we know, there are the lactose fermenters, facultative anaerobes, lactose fermenters, and they can be distinguished from Escherichia and Salmonella based on the lactose fermentation. As the non-lactose fermenters are the Shigella and Salmonella, and the lactose fermenters are the Escherichia. Now, there are 3 antigens, as we have seen, that we have the somatic antigen, we have the flagellar antigen, and there's one antigen more what we call it the capsular antigens. Now, more than 1,500 serotypes based on to the somatic antigen, O antigen. More than 50 serotypes based on to the flagellar antigen, and more than 90 capsular antigens. Combination of all these, the somatic antigen, the flagellar antigen, the capsular antigen, the K antigen, the combination, it resulted in more than 1,000 antigenic different types. This means that based on to the somatic antigen differences, flagellar antigen differences, capsular antigen differences, we have more than 1,000 species of the Escherichia coli, which are antigenically different. Antigenically different based on to the antigenicity of his capsular antigen, the flagellar antigens. So we have a huge number of the Escherichia coli, serotypes or genotypes. Now the specific serotypes which are associated with specific diseases in the humans are the O55, O11. And these 2 stains, they have caused outbreak of nosocomial neonatal diarrhea. We have another stains that is responsible for the traveler's diarrhea. We will discuss that. But the O55 and O111, that has caused the outbreaks in the neonatal diarrhea. So you never know that out of these more than 1,000 antigenic subtypes of the Escherichia coli, which one will become virulent at which time, and you can have the epidemic, or we can have the outbreak. Now the pathogenesis of Escherichia coli is very simple. If you keep in mind the anatomical structure of the Escherichia coli cell, the urinary tract infections, they are due to the patient's own colon flora that colonize the area. You see, this is very common in case of female because of the less distance between the urethra and the vagina. The organism can easily, from the colon, can easily move on to the vagina. The neonatal meningitis is because the individual, the baby, it gets this infection during the process of delivery, and it gets this infection through the birth canal, the birth of the birth canal, like we have seen in case of meningitis. The traveler's diarrhea is, of course, by the ingestion of the food and water that has been contaminated with the human feces. As you have seen, that is one of the most important public health organisms. The sewage, the fecal material contaminate the drinking water, and because it can contaminate the food material. So because of the ingestion of the contaminated food or drinking water, we can have the traveler's diarrhea. The main reservoir of enterohemolytic Escherichia coli O157 and H7. O157, somatic antigen 157, and the flagellar antigen 7. This is found in the cattle's meat. And if you eat the uncooked or semi-cooked cattle's meat, you can acquire this infection. So the main reservoir of a very specific strain of Escherichia coli, known as O157H7, it is the cattle undercooked or uncooked meat. We acquired this infection of enterohemolytic Escherichia coli strain from the cattle's meat. Now, the virulence of this microorganism O517 is because of the pili. We have the capsules, we have the endotoxins, we have the exotoxins, and we have 3 different exotoxins, right? Now, 2 of these exotoxins, they causes watery diarrhea. One of these exotoxins causes bloody diarrhea. And this is what we call as the hemolytic uremic syndrome. So, look at the structures. It has the pili, it has the capsule, contributing their virulence potential. We have the endotoxin and we have the exotoxins. So a combination of endotoxins, 3 different exotoxins, pili, capsule, really make a synergistic virulence effect, a huge damage to the human body. The 2 of the exotoxin strains, which are enterotoxins, they are watery, they cause watery diarrhea, or one of them, it causes bloody diarrhea, and this is what we call as the hemolytic uremic syndrome. So remember that hemolytic uremic syndrome is caused by the enterohemorrhagic Escherichia coli O157, and the reservoir for this organism are the undercooked meat of cattle. Very important organism or stain, and you will definitely get one of the what you call as the MCQs. Keep in mind that enterohemorrhagic responsible infecting the intestine and causing hemorrhages. These are the invasive stains, of course, because of the pili. They invade, they enter, they invade, and they cause hemorrhages and later on inflammation and all that. So E. coli 157S7 is associated with what you call as the hemolytic uremic syndrome, in which the stain involved is the enterohemorrhagic. All right. So intestinal infections continue the organism after entrance. It penetrates, it adheres to the cells of the jejunum and ileum by means of the pili, and then after you see the adhering over there and penetration, it synthesizes the enterotoxins, and these enterotoxins, they act on to the small the jejunum and ileum of ileum cells, and they cause the diarrhea. Sorry, and they cause the diarrhea. This is the cell-specific toxins, and the colon cells, they are not susceptible to this, but the cells of the jejunum and ileum, they are more susceptible. Now, the enterotoxigenic stains, it can produce the 2 types of toxins. The LT toxins or ST toxins are both of these. The heat-labile toxins and heat-sensitive toxins, either one of these or both of these. Now, we have the enterohemorrhagic stains of Escherichia coli, number 1 that we have discussed previously, and we have the enterotoxigenic stains of Escherichia coli. Enterohemorrhagic stains at the O157S7, and the enterotoxigenic stains, they have the toxins, what we call as the heat-labile toxins or heat-stable toxins. They can produce either one of them or both of these. And these toxins, they act by stimulating the adenyl cyclase. As we know that the color toxins, they also act in this way, and they catalyze the addition of the adenosine diphosphatidase. So the toxins, they are the low molecular weight toxins, are the heat stable toxins, and they stimulate the cyclase. Now the various stains of the Escherichia coli, we have enterotoxigenic stains of Escherichia coli. We have enteropathogenic or enteropathic or enteroinvasive stains of Escherichia coli. We have enterohemorrhagic stains of Escherichia coli. So please keep. Escherichia coli is one organism. We have different stains of Escherichia coli. Some of the stains, they are known as enterotoxigenic stains. Others are known as enteroinvasive or enteropathogenic stains or enteropathic stains. Some others are known as enterohemorrhagic stains. Now, the enterotoxigenic stains, they are not invasive. They do not invade into the lumen of the intestine, or they do not produce blood. They are the toxin. There is no inflammation because there is no invasion. There is no inflammation. There is no invasion of the intestinal mucosa, and it causes watery, non-bloody diarrhea. So the effect is because of the toxins, which are released by these enterotoxigenic stains of Escherichia. Now, the enteropathic or enteroinvasive stains, they are invasive. They penetrate deep. They cause inflammation. They invade the cells of the intestinal mucosa, of the large intestine, and they cause bloody diarrhea, what we call the dysentery. And we will also always see the inflammatory cells like neutrophils. So in enterotoxigenic stains, no inflammation, no inflammatory cells like neutrophils, watery diarrhea, no blood diarrhea. Enteroinvasive, invasion is there. Neutrophils would be there. Inflammation would be there. The blood diarrhea would be there. Enterohemorrhagic stains, O157, exotoxin, which is the vitotoxins. bloody diarrhea, and its effect is like the Shigella-like toxin. And this is mostly because of the consumption of inadequately cooked meat of cattle. Now, verotoxins, they attack by removing the adenine, what we call the R-ribosomal RNA, stop the protein synthesis, and it is again encoded by the lysogenic phases. So please remember three different stains of this Escherichia coli: enterotoxigenic, enteropathic or enteroinvasive, and enterohemorrhagic stains. Now we'll move on to, the E. coli 0157S7 stain. Just a few minutes more. This stain is associated with the outbreak of the bloody diarrhea following ingestion of uncooked hamburger. It's very common, very uncooked hamburger. It causes life-threatening complication, what we call as the hemolytic uremic syndrome. When the verotoxin enters in the bloodstream, it causes damage to the nephrons. And this consists of hemolytic anemia, thrombocytopenia, acute renal failure. Now, E. coli 0157S7 stain is considered to be very, very dangerous because of its syndromes, what we call as the hemolytic anemic syndrome, and that causes acute renal failure. And this stain is transferred by the ingestion of contaminated, you see the hamburger uncooked meat. Now, this it causes hemolytic anemia. There is renal failure due to the receptors of verotoxins on the endothelium, and these verotoxins, they binds over there, and it causes damage to the epithelium. There is death of the renal endothelial cells that result into the microangiopathic hemolytic anemia. They are red cells, and they pass through the damaged urea and become grossly distorted cells. And there is lysis of the cells also. There is thrombocytopenia, in which you see the platelets, they adhere to damaged epithelial cells. This thrombocytopenia is because of the deficiencies of the platelets in the blood, because the platelets, instead of being in circulation, they adhere to the damaged epithelial cells and their count decreases. The death of the kidney epithelial cell, it leads to the renal failure. And the treatment of this stain with antibiotics, particularly with the ciprofloxacin, it increases the risk of developing hemolytic uremic syndrome. So we'll have to be very careful while prescribing the drugs for the treatment of this. I think this is all about today. We will continue with this topic and we'll meet next time. Thank you very much.